So as Gautami mentioned, hello everyone. My name is Shagun Krishna, and I work as a computational and in vitro toxicologist at the Physicians Committee for Responsible Medicine. We at Physician CCRM are very much committed to utilize and promote the uses of new approach methodologies because these are human relevant, more translatable, and ethical too. And today I'm going to present my work that involved the utilization of physiological based pharmacokinetic modeling and in vitro to in vivo extrapolation. And it's a case study that basically that basically exemplify that how we can utilize the NAM-based approaches in risk assessment of or identifying the potential risk from different environmental exposure. So, and title of my talk is Understanding PBPK Modeling and IVIVE, Fundamentals and Applications in Assessing Cardiotoxicity from Environmental Chemical. You can ask your questions in the chat, and I don't know if the time permits, I would be more than happy to answer them here, or you can contact me later regarding any of the questions. So without any delay, let's start this. So first of all, I would like to thank the work before joining to PCRM. I was working at the NI.
नहीं होता HCl नहीं होता astringent नहीं होता
Okay. Before joining to PCRM, I was working at NIC Atom NIEHS, and this is my team. And I'm very much thankful to the director of NIC Atom, who was my mentor and supervisor, Dr. Nicole Klein-Stroyer. She actually helped me a lot to navigate through this space of NAMs. And before talking anything about the cardiovascular diseases, let's talk a little bit about what is high throughput screening. So this slide summarizes the high throughput screening. We call it HTS paradigm. We all know that HTS is a transformative approach in toxicology and also in the drug discovery. HTS combines automated high throughput screening. What is high throughput screening? It's basically automated technology, computational analysis, and biological assays that could be used to rapidly test tens of thousands of chemicals, and it generates a vast data sets. The ultimate goal of utilizing this high throughput screening is to uncover how chemicals, it could be a pharmaceutical chemical or it could be an environmental chemical, that how these chemicals interact with biological system, and we could identify the potential toxic or harmful effects, and we can prioritize them for the future investigation. And in my case study, in my project, we have been utilized ToxCast and Tox21 data. ToxCast and Tox21 data is a resource for the high throughput screening. So the traditional approaches to toxicity testing have posed multiple challenges for evaluating the safety of industrial and environmental chemicals, pesticides, food additives, food contaminants, and a lot of medical products. The couple of challenges include the number of chemicals that need testing, time, and resource required for the traditional animal testing, and the unexpected adverse effect that can still occur in clinical trial for drugs despite the extensive toxicological testing. So to circumvent this process, these issues, over a decade ago, the U.S. Environmental Protection Agency, EPA, National Institute of Environmental Health Sciences, NIHS, National Center for Advancing Translational Sciences, NCATS, and Food and Drug Administration of USA, FDA, they formed the Tox21 Consortium. So basically, Tox21 is a federal research collaboration that focuses on driving the evaluation of toxicology in the 21st century. And so the Tox21 researchers, they aim to develop better toxicity assessments so that tens of thousands of chemical can be quickly tested, and we would be able to know that what kind of chemical compounds have the potential to disrupt processes in the human body. And eventually it could lead to the negative health effect. If you want to know more about the Tox21 database, you can scan the QR code here and it will lead you to the Tox21 website, where you can gain more information about how the Tox21 data is generated, how it is organized, and how you can synthesize the useful information out of it. Moving along with another database that we have utilized in this study is the ToxCast. ToxCast is another very useful resource. So it is maintained by the US EPA, and So this program basically makes in vitro, medium, and high throughput screening assay data publicly available, so you can utilize this data for your research. And again, you can scan this QR code and it will lead you to the TalksCast website where you can know that how you can download the database, how you can utilize it, how the data has been generated. Next slide. Okay. So now I would like to give you a background that how I have utilized this high throughput screening, how we have utilized this high throughput screening data to identify the chemical cardiotoxic potential. So before talking about trees, let's talk a little bit about the forest. We all know that cardiovascular diseases are the leading global cause of deaths and counting over more than 17 million deaths per year. Cardiovascular system is vulnerable to several environmental exposure. Few components of these exposure have been very well characterized, say air pollution, smoking, lifestyle diseases, hypertension. But then however, there exists a potentially significant but underappreciated risk factor that is exposure to several environmental agents. We have been exposed to tons of chemical every day, like pesticides, flame retardants, polycyclic aromatic hydrocarbons, fertilizers, and different heavy metals such as arsenic, cadmium, and lead. So we wanted to see that what is the effect of these kind of environmental exposure on the cardiovascular system. And to utilize this, we have worked with the Health and Environmental Science Institute, HESI. HESI came up with the concept of failure mode. So they introduced that the cardiovascular system fails to perform its life-sustaining functions in fixed, definite ways, and they called it failure modes. And we have used this cardiovascular failure modes as a foundation for our study. These failure modes include changes in vasoactivity, changes in action potential, changes in inotropy, cardiomyocyte or myocardial injury, valvular injury, endothelial injury. And a lot of this high throughput screening assays are component of different molecular and cellular signaling pathways that could be linked to these cardiovascular failure modes. So we can map different in vitro assays to these failure modes. So we started by collecting different mechanistic target and cellular processes that are actually affecting the cardiovascular system and that we can map to the failure modes. And as you can see that we have identified as a first pass, we took all the secondary pharmacological screening target panel that most of the pharmaceutical agencies use to identify the adverse effect of the pharmaceuticals. We started with Bovis paper and they gave almost a target of panel of 40 targets. We identified those targets which could be significant to the cardiovascular system. And then we also collected several cellular processes like mitochondrial dysfunction, oxidative stress from the literature. We also queried the NCATS Bioplanet database. It's a huge resource for mapping the ToxCast and TOX21 assays to different adverse outcome pathways or to different signaling pathways. We identified a couple of assays from there as well. And then we have included several of the clinical biomarkers in the study. So we identified a list of 40 mechanistic targets and cellular processes. These are not exhaustive, but we started with this. Then we have, so the ToxCast and TOX21 data has almost 1400 assays in 10K chemicals. But not all these assays were significant were mapped to those targets. So we first start mapping the assays to those identified mechanistic target. And we had almost 414 assays across 10,000 chemicals. But again, that is a limitation of the high throughput screening assay, that it always comes with certain limitation. So not all the assays were tested. Like there were to reduce the noise in the high throughput screening data, we applied a filtering criteria. We only chose those chemicals that have been tested in the 50% of assays and the vice versa. So by applying this filtering, we identified around 314 assays that are relevant to relevant to causing the adverse CV outcomes across 1400 chemicals. The detailed methodologies has already been published, and you can go and read about it. thinking about the constraint of the time, I'm trying to skim the information as much as I can. And then we supplied this data to ToxPy. ToxPy is a ranking tool that is freely available in the public domain, and it helps you to integrate the in vitro data to rank the chemical based on their potential of the activity. It could be antagonism, it could be agonism. So by supplying our data to ToxPy, we were able to rank those 1400 chemicals as per their potential to cause adverse cardiovascular effects. And we finally had different bioactivity profile of the identified chemicals. And again, based on the ranking, we have identified several chemicals that consisted of flame retardants, that consisted of our day-to-day life chemicals like flame retardant, pesticides, herbicides, etc. We have also applied some cheminformatics approaches to identify the chemical features that are driving those bioactivity. And then we applied some use case class analysis to identify those bioactivity signatures that are relevant in that particular chemical classes. This study was published in the Chemical Research in Toxicology in 2021, and it was selected for the cover. And you can go and read this article. And if you have any questions, I would be more than happy to answer those. Now, let's come back. So now we have utilized the high throughput screening data for identifying the chemicals, but that's just the high throughput screening data. How we are going to make decisions from that high throughput screening data in for the human health welfare, or how we can utilize the human exposure component to synthesize the information out of this. So for that, we have utilized the in vitro, you have utilized multi-scale modeling, PBPK models, and in vitro to in vivo extrapolation. And then we have also utilized the human exposure data to make to to showcase that how we can utilize this in decision making. And we have also compared the results to the traditional animal test data to showcase that how these human relevant methods are better than the traditional animal testing. So let's talk a little bit about the fundamental and basics of the in vitro to in vivo extrapolation. So in vitro to in vivo extrapolation, or IVIVE, is the use of in vitro data to predict any phenomena in vivo or in human system. And IVIVE can be broken into two components: IVIV PK or TK, which is pharmacokinetic or toxicokinetic, and IVIV PD or TD, which is pharmacodynamics or toxicodynamics. So PK lets us stat what is the fate of a molecule or a chemical inside our body. It consisted of the classical ADME properties, like how it is absorbed, how it is distributed, how a chemical is metabolized, how a chemical is excreted. And it uses all the mathematical calculations. However, the PD lets us help us to know that what is the effect of that particular chemical inside the human body. Okay, so with PK and PD as our foundation, people or with PK and PD as the foundation, we can utilize different modeling approaches to predict how a chemical behaves inside the body. Basic PK models use different mathematical equations to estimate the concentration of a chemical in different body parts. And there are more complex models, which are called as compartment models. Compartment models go a step further by dividing the body into different sections with similar property. And these compartment models, they help us to calculate things like half-life and clearance of the body. Half-life means how much time that chemical is going to spend inside our body, and clearance, how that's gonna be excreted. But for a more detailed picture, we use physiologically based pharmacokinetic model. As the name suggests, PBPK means we have incorporated the human physiology behind these models. So PBPK models actually mimic the organ system, and it helps us to track the concentration of chemicals in specific tissues or in specific sections of a body. This approach helps us closer to understand how real world exposures might affect human health. And all these mathematical modelings, they serve basically as a bridge between the real world and theoretical predictions. So we are using the high throughput screening assay data, but how we are going to make the real world decisions, there comes the role of the PBPK modeling and in vitro to in vivo extrapolation. And these framework are really essential for understanding and predicting that how a chemical is going to behave inside our body and how a chemical is going, which target organs or which specific tissue it's going to affect inside our body. So it looks like a very, as I mentioned, that PBPK models are a series of mathematical equations where we utilize different parameters. And here you can see that we have utilized a compartment model. The body has been divided into different sections like lung, gut, liver, kidney, and remaining body. I know it seems very complicated, and actually it is, but you don't have to worry at all because we have a solution. So the HTTK package that EPA has built is very helpful to utilize these PBPK model in real world scenario. And in the subsequent slides, I will show that you don't have these HTTK models basically work in R, which is a programming language. But in the later slides, I will show that you don't need to be a programmer to utilize these approaches. Again, let's understand more about how PK and PD interact to influence the toxicity of any chemical. So to understand that how a chemical impact human health, it's really essential that we look at both PK and PD parameters. So as I mentioned, that PK covers that how a chemical moves through the body, its absorption, distribution, metabolism, and excretion. On the other hand, PD focuses on what the chemical does at its target sites, where it interacts with molecule like enzymes and receptors to produce certain biological responses. And together, PD and PK give you a complete picture of toxicology, of any toxicological effect of a chemical. And you can think it of this way, like that PK determines that where the chemical goes inside the body, and PD tells you that how that particular chemical is going to affect the body. And this dual approach helps us to identify that which chemical poses a real health risk. And now, and utilizing this PK and PDPB models and in vitro to in vivo extrapolation, built, we have uh, we have built a case study that helped us to identify the actual risk of the actual risk posed by these environmental chemicals, especially in terms of the cardiovascular system related endpoints. So as the name suggested, in vitro to in vivo extrapolation, you can use these approaches. You can use, this is basically that we know that a chemical is showing activity in vitro system at 10 micromolar. But how much concentration, that is the concentration in the in vitro system. But how much exposure do we need to be, how much amount of that particular chemical we need to be exposed to see that concept, to see that effect that we are seeing in the in vitro system is basically the IVIV. So IVIV helps us to translate the human exposure that we need to be exposed to get that response in the in vitro system. And there are two kind of dosimetry, two components of IVIV involved here. One is the reverse dosimetry and one is the forward dosimetry. So in the forward dosimetry, we know that, okay, we have been exposed to this much of external exposure, and how much amount do we need to see the internal concentration of that particular chemical. So imagine that we have been exposed to 300 ppm of any air pollution, and how much internal concentration are we getting using those external exposure. And reverse dosimetry, in contrast, reverse dosimetry tells us that we have been using 10 micromolar of a chemical in in vitro system and how much we need to be exposed to see that particular concentration. And now, what is the data? Can you hear me?
Yeah, Shagun, we can hear you. Can you hear us, Shagun? You've gone on mute.
Can you hear me now?
Yes, we can, Shagun.
Okay, let me again. I'm so sorry for this disturbance. Let me just share my screen again.
No way.
Okay. Mm,
Yeah, you can just turn it
it's
in
better now?
this. Yes, perfect.
Okay. And you can hear me, right?
Yeah.
Okay. So next, that what is the data that is needed to build the reliable PBPK model?
Shagun, we lost you again. Maybe you could turn off your video. Perhaps that is a problem.
It's better now?
Yes.
Okay. Yeah, let me turn off my video. Mm. It's better now, right?
Yes.
Okay. So there are basically two kind of data that is required to utilize these PBPK models. One data is related with the chemicals, that is like and these involve like the physiochemical properties of any chemical like pK log P, Henry's law constant, or absorption rate, or the clearance information. And then we require some of the human body related data, which is like the physiological properties, like what is the species that have been used, what is the body weight, what is the cardiac output, what is the tissue weight and volume, and such, and so on. And okay. Mm. Oh my God. Okay, can you see my next slide now?
Yeah.
Okay, it was stuck for a moment. I'm sorry. So this is where, as I mentioned you, that I'm gonna let you know in the latest slide that you can utilize all these complex approaches in a very easy and convenient format. So NIC Atom has developed a dashboard, a set of toolbox, which is called the Integrated Chemical Environment. And this ICE dashboard is really very helpful if you want to utilize the ToxCast and TOX21 high throughput screening data, and if you want to utilize all these tools. So as you can see here, like there is a search tool that helps you to search any of your chemical, that whether or not your chemical of interest is present in the ToxCast and TOX21 chemical, ToxCast and TOX21 data. And then comes the chemical quest tool that helps us to know more about your chemical by identifying the similar chemical. You can define the similarity threshold in the chemical quest tool, and it will let you know all the neighbors and all the data related with your chemical. Again, the high throughput screening assay data is a complex data, and there are several ways that how you can fit the different concentrations in the dose response curve. So there is this curve surfer tool that helps you to identify those, that helps you to make decisions, or that helps you to understand more about the dose response curve of those chemicals, of any chemical of your interest, or a set of chemical in a particular assay. And PBPK tool helps us to utilize the PBPK models through the HTTK, basically through the HTTK model. So HTTK models have been implemented in this tool. And then in vitro to in vivo extrapolation helps us you to do the IVIV analysis, and it gives you the dose that is required to be that is required to be exposed to see the effect that you are seeing in the in vitro system. chemical characterization tool help you to categorize your chemical based on the use case classes on the basis of consumer product database of the EPA. And you can scan this QR code and it will lead you to the website of the ICE dashboard. And this is how the ICE PBPK, the output of ICE PBPK tool looks like. I particularly like it, and I encourage all my summer interns and my mentee students to use this because it gives you publication ready. I mean, like, one of the wonderful feature is that it gives you the publication ready images. You don't have to tweak these images. You can use this. These are like really high quality publication ready images. You can just use them in your reports or in your sometime in your publications as well, or in your thesis, or in any document that you are working on. So as you can see that here in this, you have to provide your parameters and like the chemical information and the required parameters, and it gives you the concentration of that particular chemical in different body parts. So it gives you the concentration in liver, concentration in kidney, concentration in gut, lung, and rest of the body. And not only this, I believe that the recent ICE dashboard does include the updates, and now you can even utilize the fetus model as well, placenta models as well. And this is the output of IVIV tool. So it allows you to estimate the dose that is called as equivalent administered dose that would result in the plasma concentration of a chemical that equals to the active concentration in any given in vitro assays. Not just in one assay, you can even use a set of multiple assays for as an input for this tool. And it also helps you to contextualize the dose predictions against the in vivo assay data and human exposure prediction. So if you want to see that how your predictions are against the traditional animal testing data, you can do it right here. You don't need to use any other script or any other programming language to do all that analysis. And the recent update even include that you can also give your custom data, like the output of the in vitro assays that you have been doing. I mean, like, you can use the ToxCast and Tox21 data, but if you have your own data, you can use that also. You can just put that as input and it will give you all the results. And again, if you are wondering that how you can utilize this ICE tools in your study, you don't have to worry. PCRM has come up with a very wonderful and very nice training. I think 2 or 3 years before, you can scan this QR code and it will lead you to that training. It was a intensive 3-day training workshop where the ICE developers have demonstrated that how you can utilize this tool in a comprehensive way, not just the tool, but also the theory behind those tools. So I would highly encourage anyone who wants to use the ICE dashboard, go through this training. And if you have any information, if you have any questions, you can also reach out to us. And again, PCRM, if you want to know more about all these NAM-based resources, that how you can utilize the high throughput screening data, how you can utilize PBPK modeling or any other organ-on-chip models, especially in the regulatory setting, we have a learning management system that is called as NEURA, NAM used for the regulatory application. It has a collection of several webinars and trainings related to the NAM use. You can register to any training and you can get certificates as well. And if you are working on a topic and you feel that a training is not available for that particular topic, please do reach out to us. We are more than happy to accommodate your request. And that's the email, neura@pcrm.org. And if you will scan this QR code, it will lead you to the Neura Learning Management System portal. Okay, so now we have seen theoretically that how we can utilize the in vitro data to make decisions and to incorporate human exposure. So we have identified the chemicals that are most concerning cardiotoxicity profiles, but then assessing the actual risk posed by these chemicals required us to translate the in vitro finding to real world relevance. So this is where we have utilized the IVIVE extrapolation. I know this slide is pretty busy, it's overwhelming, and I'll try to break down the component of these slides into component of the slide into several small parts in the subsequent slides. And this basically, this is the workflow which shows that how you can utilize the PBPK and IVIV approaches in identifying any chemical of interest for any endpoint that you are working on. So as I mentioned that we suggested with chemical assays, can I use the pointer somewhere in the Zoom? Oh yeah. Oh. Okay. Yeah. Oh, I'm so sorry. I thought that I have been using it since long. So as you can see here that we started with chemical assays from ToxCast and Tox21. We had cardiovascular failure modes. And then we built the cardiotox pie to rank the chemicals. And then again, the biological activity span across several assays, but we want to be very conservative in our approach. So we just took the most sensitive activity, that was the minimum AC50 of the chemical in that particular set of assays. Again, we knew that high throughput screening data comes with a lot of uncertainty. So to circumvent those to address that issue, we have incorporated the uncertainty by utilizing the ToxBoot package. That's another package developed by EPA that helps you to identify the upper and lower bound of any in vitro bioactivity. And then again, we have used the ICE dashboard. By that time, ICE, I mean, like when I was doing this study, ICE doesn't have the PBPK and IVIV tool. So I basically use the HTTK R script, but now you can utilize all those approaches in a way more user-friendly manner. So I have utilized the ICE dashboard to do I have utilized the HTTK scripts to do the in vivo and in vitro to in vivo extrapolation. And then the dose that we got by applying the IVIV, we have compared those with the traditional animal study experiments to see that whether or not the utilization of these NAMs are giving us better result than the traditional animal study. And to make more human relevant decisions, we have also utilized the human exposure data that was available to us via Tox, oh gosh, via database. Oh my gosh, why I'm forgetting the name? It's okay, I'll let you know in the. So the animal data was available to us via ToxRev, and this was available to us via ToxCast. No, not Tox, ExpoCast. Yeah, I'm so sorry. Yeah, so ExpoCast database lets us, it's again, all these databases are available in the public domain and anyone can use it. So ToxRef is the database for the traditional animal studies, and the ExpoCast is the database for the exposure monitoring of these different chemicals. It has the real world data which has been developed through enhanced database, but other than that, it also contains the prediction of the human exposure data via scene 3 models. And by utilizing these, by comparing our estimated doses to these different data, we have derived different safety metrics: POD ratio, bioactivity exposure ratio, and margin of exposure ratio. And with the help of, by analyzing those results, we have identified several chemicals that have been found that were not harmful in the traditional animal studies, but were found harmful utilizing our in vitro data. These are few examples of those chemicals. And based on the human BER estimates also, we have identified a couple of chemicals for which our study reveals that we have been exposed to much higher amount than we should be. And again, I wanted to bend, I wanted also to see that what are the different, if you recall, I mentioned that there are 3 types of models, very basic models. Those are the one compartment model, which just takes the whole body as one component. And then there comes a more complex model like PBPK model. So I wanted to see that what is the how does the choice of using these different models affect the results? So for those reasons, I have utilized different PBPK models, 1 compartment, 3 compartment, and PBTK. I also wanted to see that what is the effect of these chemicals in different time, in different duration, and in different dosing scenario. So we simulated them for 3 dosing scenario, 3 day, 24 hour. I mean, like, how much effect will you see if you have been exposed to that particular chemical for 3 days at 24-hour interval? We also wanted to see that if you have been exposed to multiple times for a particular chemical, what would be the results? So for those reasons, we have utilized 3 day, 2 hour interval. And we also wanted to mimic the long-term simulation. So we have utilized the 30-day, 24-hour interval. Again, we had 3 estimates of bioactivity, minimum AC50, upper bound, and lower bound. And we have utilized the 2 dosing scenario. You have been either you could be exposed by taking any chemical orally or IV. And then for the XPOCAST data, we have utilized 2 data points, 95th percentile and median exposure. And for the comparison of the animal data in vivo PODs, we have utilized the ToxVal database, and we have utilized 2 measures of the POD, minimum POD and 5th percentile of POD. And these are the equations. By utilizing these equations, we have derived the 3 safety metrics. So the POD ratio was defined as the difference between the log 10 POD in vivo animal study values and IVIV derived EAD values. BER is defined as the difference between IVIV derived EAD values to the human exposure values, and MOE was defined as the difference between the POD in vivo animal study values and the human exposure values, which were obtained from XPOCAST database. And so how do you interpret these safety metrics? So a negative log 10 POD ratio indicate that the EAD that we have derived from the IVIVE is greater than the POD of the in vivo studies or animal doses, meaning that animal study provided a more conservative doses. However, if wherever we were getting a positive POD ratio, that means that the EAD IVIV was lower than the traditional animal study data, and the in vitro data was more conservative. Similarly, a negative BER indicate that the EAD XPOCAST is higher than the EAD IVIV. That means that indicating that there was a overlap in the predicting doses where cardiovascular relevant activity is observed and where human may be exposed. And finally, a negative MOE indicates that the EAD XPOCAST is higher than the POD in vivo. Okay, next. This is the figure where we have provided the distribution of the exposure data, EAD, IVIVE, and POD in vivo. So A is where we have the positive POD ratio values. So for these set of chemical, animal studies were not conservative. The human relevant or the NAM based IVIVE or the NAM based or the IVIV derived EAD was more conservative. And similarly, in this figure, we have provided the exposure comparison from the XPOCAST estimates and the EAD that has been derived from the IVIV study. Again, we have utilized a different dosing scenario, but I'm just showing you the results of 3 day, 2 hour, because that was the most conservative dosing scenario that we could achieve. And in this study, a total of 92 chemicals had BER less than 0. That means that for a set of 92 chemicals, the potential for exposure to occur within the bioactivity dose range extrapolated from the in vitro data. And remember that these BER in the log values. So if any chemical is exhibiting BER -2, it means that the estimated human exposure level is approximately 100-fold higher than the dose associated with the CV, with the cardiovascular perturbation on a molecular or a cellular level. So this implies a very low margin of safety and suggests that these chemicals may pose significant risk and we should investigate them further. And as you can see that there are a lot of chemicals that we see in our day-to-day life, like naphthalene, a lot of phthalates, a lot of estradiol chemical, of course, a lot of BPAs. And again, I couldn't put those two figures in one slide, so I just decided that I'll break those in two figures. So again, this is again the chemicals that were showing negative log BER. Okay, and this is the characterization of chemicals that cause adverse EV effects. I told you when I was describing the ICE that we can also utilize the ICE dashboard to categorize our chemicals, that what is the spectrum of those chemicals in our day-to-day chemical products that we use. So as you can see that a lot of these chemicals have been found in personal care products, in pesticides and herbicides, in flame retardants. Many of them were component of industrial chemicals and plasticizers. Of course, a lot of forever chemicals or PFAS-like chemical and household and cleaning products. And then I took the literature, I just like searched them in the literature to see that whether or not these chemicals have any known CV effect, and these are the known CV effect that I could found. And again, we also applied another NAM-based approach, which is called as geospatial mapping. So now we have identified that, okay, these are the chemicals that are exhibiting high in vitro activity or that are exhibiting potential activity in vitro system. How we can utilize by utilizing the IVIV, we have identified that, okay, these many chemicals are relevant for should be further investigated because of the comparison with the human exposure. But then we wanted to see that what is the regions where people might be affected by cardiovascular risk of these exposure. This is a very I would say that this is not an analysis that we could use in the regulatory system, but this is another example that how we can utilize these approaches to address different different issues posed to human health by these environmental chemicals. So as you can see that it gives you, by utilizing this approach, we have identified the regions within the United States that could be potentially rich for the exposure to these chemicals. And this can help us to make different kind of decisions, but not just alone. So that is another, I would say that this is just an example that how we can utilize the different NAM-based approaches in different scenarios to address different health issues. And now the conclusion. So we have, by utilizing this NAM-based workflow, we have identified several chemicals with cardiotoxic potential at human relevant exposure level that may not be flagged in any of the traditional animal studies. By comparing the bioactivity derived doses with human exposure estimates, we have identified several substances that have been commonly used in our day-to-day life, underscoring the limitation of the animal models in capturing those human-specific effects. And again, geospatial modeling in this study, it highlighted the regions where the community may face high cardiovascular risk. And these are the take-home messages. So NAM-based new approach methodologies, I have utilized this, I've used this term 1,000 of times. So NAM integrates that NAM integrates the high throughput in vitro assay and computational models to predict cardiovascular risk directly to human biology. By utilizing the bioactivity exposure ratio, it gave us a human relevant metric by comparing the in vitro bioactivity doses with the actual human exposure. POD ratio helped us to benchmark the human relevant doses against animal study threshold. And combining these NAM, IVIV, and geospatial modeling, we can utilize this whole case study as a NGRA example case study as well, which is next generation risk assessment. And with this, I would like to conclude my talk, and I would like to thank the director of NIC Atom, Dr. Nicole Klein-Stroyer, and my colleagues at NIEHS, Shawching, who actually helped me to do a lot of the PBPK modeling stuff. And Kristen and Kyle actually did the geospatial modeling component. I'm not an expert in that component. And I would also like to thank my supervisor at BCRM, Elizabeth Baker. She has been very supportive throughout this short time at PCRM. And I would also like to thank CPHSMS people and all of you for patiently listening to my talk. And now I would like to welcome any questions.
Thank you, Dr. Krishna, Shagun. That was a very good talk. And thanks to you, we got to know a lot about these models. So before we move into the Q&A session, for those people who are attending the webinars right now, we have put in a link to give your feedback so that we can improve our webinars. And also, if you want any kind of information on CPHMS activities or updates, you can also access us through other link. It's in the chat box. Over to Kasturi.
Yeah. Thanks, Shagun. It was an extremely interesting talk, and we already have a few questions in the chat box. So perhaps I could read them out and we could answer them. So
Yeah, sure. Can I stop
asks,
sharing my screen now?
yeah, sure. So Megha asks, how can the timescale differences between the in vitro data and in vivo data be incorporated into the PBPK models to improve the predictive accuracy?
That is what, like, if you want to simulate or if you want to see the chemical for like, you can, like, that is what ICE lets you to do, or any HTTK model lets you to do. If you want to see the chronic effect, if you want to see the subchronic effect, if you want to see the acute effect, you can utilize the different time intervals based on your interest to see that how the models are different. And then if you want to compare, you can compare by utilizing these different time frames, you can compare the results. Does that answer the question?
I think Megha may answer.
So, okay.
So we'll wait to hear from her. In case, Megha, you would like to ask something again, do unmute yourself and feel free to ask another question. So there's another question from Dr. Mamta Mishra, and she asks, after having data on in vitro cell cultures, how long does it take to get an IVIE conclusion?
Okay, it depends upon your will and how much you are committed to do it. I would say that, of course, whenever you start a new work, it will take some time, but again, there are resources available. So it's not very much time consuming, but yeah, there are like different components. You have to collect your data. You have to, if you're a first time user, you have to learn a little bit more about what are the basics, and you should be knowing that what answers you are looking for. So I don't think that there is a certain time frame, but yes, I would love to emphasize that it's time and cost effective. So it won't take like years to do these calculations. If you are ready with your parameters, if you're ready with your questions or what you are looking for, it won't take, like, performing a calculation doesn't take much time. For me, performing a calculation for 800 chemical for 30 days, it just took some minutes.
And so if someone has particular questions on this, on their research aspect, can the training that you talked about earlier answer some of these questions? Could these questions be asked in the discussion aspect of these training sessions, perhaps?
Yeah, of course. If we organize any subsequent training, I would encourage all of you to come up with your questions so that I can answer them real time.
Perfect. Yeah. Alongside, Dr. Mishra asks, how many databases or softwares are required to get an IVIE conclusion?
Okay. So again, like there are basically two kind of data, one related with the chemical, one related with the in vitro data. If you have your in vitro data, then you can use that in vitro data. If you don't have, then you can look your chemical in the ToxCast and Tox21 database and you can identify that. So that is one kind of data. For for calculating the chemical properties, either you can look for the experimental parameters. If the experimental parameters are not available, you can utilize the opera, again, which has been developed by Dr. Kamal Mansouri, who is working in NIC Atom. He developed the opera, and utilizing the opera, you can calculate all the physiochemical property needed for any chemical to do the IVIV analysis. So that is one thing, and then you just need to use the eyes. That's what I would say.
Okay, great.
Mhm.
Thanks. So I don't see any more questions. I'd like to
Okay.
close with one question that I had.
Uh-huh.
In your observation, do you see a lot of Indians using these softwares? Do they publish with these using these databases? What has been your observation in general?
It has been used a lot in the regulatory agencies. Even we are working very closely with the OECD, which is Organization of Economic and Corporation Development, that consists of 38 countries. A lot of regulatory agencies, OECD, ECA, even US EPA, has been using these kind of new approach methodologies to make the regulatory decisions. So yes, I would say that I've seen a lot in United States and in European countries, they have been using it. If you're asking about like context of India, then I apologize, I don't have much information about that.
Yeah, perhaps we could have an information session with Indian regulators as well
We would
to,
love to do that.
to
Yeah.
train them on how to use these.
Yes.
Okay, great. So thank you once again, Shagun. It was a pleasure to have you.
Thank
And
you very much for having
I'm
me.
sure many participants will write to you about any queries they have.
Thank you very much, Kasturi, for having me. And thanks to all your team, Tejasvi, Gautami, you, and Dr. Surat. You all have been doing a great work. Thank you
Thank
very
you
much.
so
And
much.
thanks to the
See you
audience.
again.
Mhm.
Bye.
Bye.